This is Eeva Leinala from the OECD, and I thank you for joining our webinar today. This webinar is organized in the context of the OECD UNEP Global PFC Group that was formed to help support the SICEM resolution on shifting to safer alternatives for PFASs. The group has a number of initiatives, but one includes a webinar session on different topics. So it's great that you can join us today. And one area that the group wanted to explore more was analytical and monitoring methods for PFASs. And so that is what the webinar today is about. And in fact, we will have a second set of presentations on the same topic, likely in September as well. So there's a lot of interest and different suggestions of different speakers. But today I'm very pleased to have three speakers with us. Shoji Nakayama from the Center for Health and Environmental Risk Research from the National Institute of Environmental Studies in Japan. This will be followed then by a presentation by Chris Impellitteri from EPA's Office of Research and Development. And then finally, Stefan Posner, who's an independent researcher, but who will present work that's being done within CEN and ISO on the development of test methods for analysis of PFAS in textiles and leather. So each of the speakers will have about a 20-minute presentation, the three speakers. And then I'd like to ask Shoji to go ahead and share your presentation.
Okay, so first of all, I would like to appreciate the OECD to provide me with such a fantastic opportunity to talk about my sort of life work, about PFAS and this webinar series. I am going to talk about the worldwide trends in the method development for the analysis of PFAS in the environment. I just like to acknowledge my co-authors, and here I listed here, as well as my old friend from US Environmental Protection Agency. I stayed there for 6 years, and without them and Andy Lindstrom, Mark Striner, and Mark Mills, I am not here. And of course, Chris, and I'm so happy to have this opportunity to make us reunited. I feel world is small. But anyway, so here's the forever chemicals facts. Poly and perfluorinated substances, shortly PFAS. This is a plural noun, but they consist of a vast cosmos of chemicals with one or more perfluoroalkyl moieties. I have the picture of Adona here, which is in relatively new compounds, PFAS compounds. And then more than 4,000 PFAS are currently on the global market, according to the CAS registration. And PFAS are widely used and distributed everywhere. PFAS are used on our daily, the products of daily use, as well as some industrial, products. And then also PFAS are used in like a firefighting forms. So they are everywhere. And also PFAS have been used widely and for long term. They distribute in Arctic or Antarctic areas too. And some PFAS are persistent in humans in the environment. But recent substitutes are aimed to break down faster and to be eliminated quicker, but still they're persistent. And we're also exposing to PFAS almost constantly. So that means even though they are short-lived, but they are there every time. And some PFAS have been found toxic, I would say some, but to both humans and ecosystem. But perfluoroalkyl moieties are so beneficial to humans that they would not be retired from the market very soon. And then research, especially in environmental health, is not sufficient at this point to fully unveil PFAS impacts on our health and the ecosystem too. So I would say PFAS are chemicals that demand forever research. And here I listed some known PFAS, with their chemical structures. We have perfluoroalkyl groups like carboxylic acids, sulfonic acids, phosphoric acid, phosphinic acids, and also iodides, and fluorotelomer groups, which means there are some CH2 or CH2CH2 moieties in between the perfluorinated moieties and some acidic moieties. And there are some perfluoroalkanes, alkanes, and sulfonamides, and sulfonamido acetic acids, and sulfonamido ethanols. And there are some perfluoroethers, sulfonic acids or carboxylic acids, which is HFPO DA, or more commonly called, it's a Gen X, which is a product name. And there are fluorotelomer olefins, alcohols, iodides, and there are much more complicated, structures too, like a phosphate diester or triester, or cyclic perfluoroalkyl acids, fluorotelomer alkylates, metacholates, and some phosphate esters too. So we're talking about a diverse kinds of compounds here. So I will summarize the current method development status here in this slide, and then just briefly mention about it. So there are sampling and extraction cleanup steps, and then followed by instrumental analysis for the PFAS analysis. For the sampling methodologies, we have been doing manual sampling and for some large volume sampling, but now the small volume and the online analysis is developing. And we call that could be green chemistry, so we can save solvents as well as other and energies too. And for the extraction and cleanup steps as well, we've been using liquid or ion pair extraction in the first decades of the research, but it moved to solid phase extraction or solid phase micro extraction, and then now evolving technologies like Voltex assisted or dispersive liquid micro extraction, or multiple, or other SPME, or those kind of things to do green chemistry too. Also, we are having more sensitive instruments like GC-MS and LC-MS, high resolution MS, or we can do GC by GC or LC by LC, high resolution or mass spec, for the non-target analysis. And online solid phase extraction, LC-MS is getting more and more common to save, to have a smaller volume of samples, as well as to save solvents and other materials for the extraction and cleanup steps. And then there are some non-target methods development too. I'm going to talk about this later in detail. But those technologies are still in the stage of the infancy. So we do need more method development in this kind of field. So first I'm going to talk about air samples, which is kind of the samples that are analyzed in the very first stage of the PFAS research to reveal the fate and transport of the PFAS compounds. So for the air samples, target PFAS are like volatile and neutral PFAS mainly. For example, fluorotelomere alcohols and PFASs and PFASs, and esters and ethers, using the PFAS PFAS type of collection, or solvent impregnated polyurethane form, which is like a U4 type of sampling method. And for neutral PFAS, SPE cartridges are also used. And then air volumes to collect are typically large, but it's been decreasing in the recent years. And SOx-ray extraction of the PFAS samplers is common. But for the SPE, the small amount of solvents are being used. And GCMS has been used for the neutral PFAS, but LCMS has been used for ionic PFAS analysis. And here I list some results. So there are global monitoring campaigns, and then they revealed PFAS or PFAS precursors like fluorotelomere alcohols are everywhere in the world. And then there are considered to be a cause of the long-range transport of the PFAS compounds. And then there's some research for the indoor air samples, and then the concentrations tend to be higher. Here, for example, in China and also in Norway, they are much higher than in atmospheric concentrations. So there could be some sources indoor or near the houses. But some bioindicators, such as vegetation samples, using the vegetation samples like a tree leaves or bark, may become some important alternative tools for analyzing the atmospheric transport of PFAS. And next, I'm going to talk about the aqueous matrices, such as drinking water, ground and surface, and seawater and wastewater samples. And those samples have been analyzed widely in the world. And then target PFAS includes carboxylic acids, sulfonates, and phosphor and phosphine, and fluorotelomere sulfonic acids, and fluorotelomere alcohols, and other emerging PFAS, like Gen X and donor, and then other things. And grab sampling techniques has been used widely and for a long time, using high-density polyethylene or polypropylene containers. Sometimes they use glass containers, but they tend to use plastic containers to avoid sorption. But still, you have to rinse the sample containers with the methanols or other kinds of solvents to recover the PFAS absorbed on the sample containers. And typical sampling volumes are like liters or half a liters, but it's getting smaller and smaller in recent years, using like online SPE type or direct injection type of techniques. So as I listed up here, the solid phase extraction is a major way of the extracting and cleaning up the PFAS in the aqueous matrices. But recent years, the people are going green chemistry, and so online SPE methods getting common at this point. And mainly LCMS has been used with the electron spray ionization, but some people use high resolution mass spec and then other ionization techniques other than ESI. And sometimes they use GCMS or MSMS, but mainly LCMS is used for the PFAS analysis and aqueous matrices. And I listed some results and recent results from the world. For example, the surface water or seawater, they tend to find PFAS in low concentration, but in China, some of the PFAS like PFAS and PFOA are found to be very high concentration, and even though in recent years. And in the wastewater treatment plant in Australia, they found carboxylic acids and sulfonates in relatively lower concentrations. In tap water, the concentrations are typically low, but in South Korea, there are some higher concentrations were found. And also in the groundwater, the concentrations are typically lower, but in China, there are some high concentrations also found. And especially, they found a new PFAS species like chlorinated PFASs in China. And those are the first discovery of a new species of PFAS. And then some people, here's a new PFAS species. They looked at those species in the world, and then also found them in the world surface water system. So the major development in the recent years for the aqueous matrices is the miniaturization of the extraction procedures. So they are going green chemistry. And also they found a new PFAS in the surface water. So my friend Mark Striner first identified Gen X, and then some Chinese people also finding a new PFAS in their surface water system. And then going to solid matrices like sediment, soil, sludge, dust, they found many kinds of PFAS, especially the precursors to the perfluorinated carboxylic acids or sulfonates, like PAP and SAMPAP and Adona, which are more complex precursors. They mainly use stainless steel grab samplers or button samplers and hand trawls or knife to collect samples. The sampling volume is typically smaller, and then they also collect house dust, the 100 milligrams to grams of house dust. And extraction and cleanup methods are mainly they use liquid extraction followed by some SBE cleanups, and then followed by GCMS, MS, or LCMS analysis. And here I show some examples of the concentrations found worldwide, and some PFAS and PFOA were found in sediments worldwide, also sludge, and also soils. And there are some worldwide soil monitoring project done, and then found PFAS everywhere. And also PFAS were found in house dust. Some cases in the very high concentrations. So, and also the PFAS tend to exhibit a strong absorption to solid matrices. So we have to have more efficient extraction methods for diverse classes of PFAS. And the house dust is one of the most important routes of human exposure. So a method suitable for both volatile and non-volatile PFAS in house dust is warranted. So I'm going to move to the wildlife and humans. There are many kinds of samples were analyzed, including fish, frog, eel, marine organisms, and eggs, and every kinds of wildlife samples, as well as blood, urine, nail, hair, and many kinds of human tissue and liquid samples. And target PFAS a wide range, including PFCAs, SAs, and other kinds of samples, especially phosphinic and phosphoric acids. And others include more like precursors and some emergent PFAS compounds. And samples are typically collected in plastic containers. And then typical volumes are 1 sub-grams to grams tissues, or microliters levels to milliliter levels, and some tens of milliliters of samples for urine samples. And then SLE and LLE, or liquid or solid liquid extraction, ion pair extractions has been used widely, but to extract more PFAS from the protein-bound PFAS, the alkaline digestion methods are typically used. And then also for the human samples like blood and urine, and online SPE samples, sample extractions are widely used. And the LC-MS are typically used, and then most recently, online SPE LC-MS has been used widely. And here are some results. You know, they've been finding PFAS in every types of wildlife, like including beluga or crocodiles, or even polar bears, and that means they are everywhere. And then also human samples tend to show relatively low concentrations to very high concentrations, depending on where they live and what they consume, what the lifestyle they live. And then there are many kinds of PFAS are found in human samples, including including legacy PFAS, as well as novel new PFAS, like here, monopap, dipap, or phosphinic and phosphoric acids, and then very new PFAS species. And especially in Asian countries, where most fluoropolymer production is now carried out, it is important to measure PFAS alternatives. And then over 3,000 PFAS belonging in 10 different classes are found in China. So it is very important to develop a new comprehensive methods to capture those kind of things. And then non-target and non-specific analysis are widely being used to find as yet known PFAS. So there are some new methods coming up, like a combustion ion chromatography, or total oxidizable precursor methods. But there's a non-target analysis is still infancy stage, so we need a method development. And a vital shortcoming of non-specific methods is that their results cannot be used for estimating toxicological effects. So we need to have a method to bridge non-specific analysis and toxicological evaluation. So here, this is my last slide, and I have the challenges, the current challenges and the future directions. We have ever-growing list of PFAS, and then and sometimes not fully understood. So we need a new and sound management scheme. And then so some of the countries like EU put in a new framework. So we need some a new legislation or regulation or a monitoring framework to capture entire PFAS picture. And there's still little known about the effect of most of the PFAS on humans and ecosystem health. So I don't know how, but we probably need more studies, and we need some computer-based effect modeling, or maybe a development of the toxic equivalency factors for PFAS to comprehensively capture the effects. And also we need some statistical models development. So like capturing the mixture data, and also we have many left sensor data, how we can sum them up. That's a problem. So there are some new statistical methods are being developed. So we still need to focus on this kind of data handling methods. So with that said, I would like to conclude my presentation, and then I would like to hand the podium over to my friend Chris.
Excellent, thanks. Hi everybody, my name is Chris Impellitteri. I'm with the United States Environmental Protection Agency Office of Research and Development. I'm the Associate National Program Director for the Water Research Program within ORD. And I'd like to recognize my colleagues Mark Mills and Andy Gillespie, also in the Office of Research and Development, and their contributions to everything having to do with PFAS and our research and results and analytical methods. First, a very quick disclaimer that I'm required to put in here. It essentially says that the EPA does not necessarily agree with the views I'm about to present, but I don't think there's anything too controversial here, so I don't think there's going to be a problem. So I'm going to jump right into the methods. Shoji gave a very good overview of PFAS and the types of PFAS that we're looking at throughout the world. Roughly, I'm going to divide methods into targeted, as Shoji briefly described in the previous presentation, and non-targeted methods. Targeted methods, you're looking at very specific chemical species. Once the analysis is complete, you can't really go back and look for other analytes. Non-targeted methods use high-resolution mass spectrometry. You can try and quantitate any of the unknowns that you find using non-targeted methods if laboratory standards exist, and that is a big if, because we're talking about a very large number of compounds. You can screen for known suspects, and you can also look for new or unknown analytes. So Shoji mentioned in the previous discussion a compound that's commonly referred to as Gen X or HFPODA. Non-targeted methods were used in finding that particular compound and elucidating its structure. This is not a very straightforward method. It takes a lot of talent, a lot of education, and a lot of art, if you will, when you're looking at non-targeted method data. But it can be a very powerful tool to identify compounds that you may have in your sample that you don't know about. So very briefly, non-targeted analysis, you're essentially using mass spectrometry to assign a high-resolution mass or mass-to-charge ratio for peaks that you observe in your chromatogram. You can use software in some instances to calculate the exact number and type of atoms needed to achieve that mass. And then you use the instrument to essentially break the molecules apart and analyze what the results are of that fragmentation. And using the masses of both the original and the broken down products, you can go back and put things back together and start assigning formulas and structures. And then again, as I mentioned, it's always best if you can obtain a commercial standard to go back and confirm that identification of that unknown if you can. So I'm going to also broadly classify three types of methods. There's EPA standard methods. These are methods that have been through multi-lab validation, and they support a particular regulatory or guidance effort. There's research methods that we have. These have been developed typically in ORD or EPA regional laboratories for research purposes. They've gone through quality assurance. They've perhaps been peer-reviewed via publication, but they have not been formally multi-lab validated. And thus they are not considered EPA standard methods. And then lastly, developmental methods. And these are really methods that are currently undergoing research and development and aren't quite at the the publication or QA level. Furthermore, there's 3 broad categories of EPA standard methods related to water and solids, and that's really what I'm going to focus on during this brief presentation. The links are there if you want to learn more about the different protocols for these different methods. So there's Safe Drinking Water Act methods, Clean Water Act methods, and SW846 series methods. First, I'm going to talk about drinking water methods with an EPA. The first drinking water method that came out was Drinking Water Method 537. That has undergone a first revision as of November 2018. The original 537 method was updated to include 4 additional analytes, and those 4 additional analytes underwent a multi-laboratory validation. The revision also incorporated some clarifications that were issued in an EPA technical advisory. It is an LCMS-MS method using internal standards, and the lowest concentration minimum reporting level ranges are 0.53 to about 6.3 nanograms per liter. The Drinking Water Method 537 within the United States and perhaps outside the United States has been quote unquote modified quite a bit. And we always like to stipulate that Method 537 is if it's the modifications that are made by a commercial lab are outside those that are explicitly listed within the method, it's no longer considered 537 by EPA. So you hear commercial and some contract labs say, well, we used a modified 537. Well, it's not. If it was modified outside of the method itself, it's that laboratory's SOP, and they could perhaps say it's based on 537, but it's not really a modified 537 unless those modifications are outlined specifically in the method. By far the most common modification is the inclusion of isotope dilution to calculate final concentrations, and that is outside of the scope of 537.1. A relatively new drinking water method that's been developed is Drinking Water Method 533. This uses solid phase extraction and isotope dilution, and it targets generally PFAS that are less in length than 12 carbons in the chain. And one of the main reasons it was developed is because 537 generally performs poorly for very short compounds, C4s, C5s. It uses solid phase extraction, LCMS-MS isotope dilution, and it will support the 5th upcoming unregulated contaminant monitoring rule. Without bogging things down too much and going into too much detail, the unregulated contaminant monitoring rule requires that EPA work with drinking water systems throughout the United States to monitor for unknown chemical and biological contaminants every 5 years. The method itself was released in December 2019, and there's a link there provided for you to access that particular method. This slide simply shows a summary of the PFAS compounds that are covered by Method 533 on the left-hand column, by both methods, 537.1 and 533 in the middle column, and those compounds that are only included in 537.1 on the right. The bold font compounds in the middle column were analytes that had been on a previous unregulated contaminant monitoring rule over 5 years ago, where we had 6 compounds. The next unregulated contaminant monitoring rule monitoring period will probably be in late 2021 or early 2022. And I can reasonably guess that there's going to be a lot more PFAS compounds during this next monitoring effort. So let's move on to non-drinking water methods, but aqueous matrices. We have developed and multi-lab validated a direct injection method that's under the SW846 series of methods. This is method 8327. It is not for it's not for drinking water. The method was validated for groundwater, surface water, and wastewater effluent. It's a very simple method relative to solid phase extraction or some other form of extraction in isotope dilution. We focused on the simplicity and the robustness of the method to really try to maximize throughput for production lab use. This method, since it's a direct injection, minimizes sample transfers and extractions and extra filter steps and chemical additions, such as pH adjustments. The link to the method is on this slide. Feel free to access it and to download the method. The method includes 24 PFAS. They're all commercially available standards that include both the native and isotopically labeled standards. So the isotopically labeled internal standards are simply used to calculate your recovery of that isotopically marked standard. It's a direct injection method, as I mentioned. It was developed in Region 5 in the Chicago Regional Laboratory. It's very similar to the ASTM method D7979. They're both based on the same standard operating procedure. The multi-lab validation was completed in 2018, and the Office of Land and Emergency Management that oversees the SW846 process is currently finishing addressing public comments. The comment period closed in August of 2019, and they hope to finalize in March 2020, right about the same time that everybody was being sent home and laboratories were being closed because of the pandemic. The method is shooting for 10 nanograms per liter as far as detection limits goes. And it also has an associated preparation method for aqueous matrices, and that's SW846-3512. Just a quick note about that target quantitation limit. As Shoji mentioned, manufacturers are coming up with better and better instrumentation, it seems like, every day. And right now, some of the newer instruments can run this method and easily achieve 1 nanogram per liter using the direct injection method. In addition to the direct injection method, which has been multi-lab validated, we are currently single lab validating and hopefully following that with a multi-lab validation on a non-drinking water isotope dilution method for both water and solid matrices. So this isotope dilution method will probably be more robust for really complex matrices, things like wastewater influence and landfill leachates. By using the isotope dilution, we can hopefully account better for matrix effects such as absorption through using the isotopes and calculating final concentrations based on isotopic standard recoveries. We're working very closly with the Department of Defense on this, and we hope that folks that will want to use a more, or maybe do a deeper dive, they can use 8327 to screen samples prior to using a more detailed isotope dilution method. It includes 40 PFAS analytes and includes all of the analytes that are listed in 537.1, 533, and 8327. It focuses on assessing PFAS in surface water, groundwater, wastewater, landfill leachates, but also solids, including soil, sediments, biosolids, and tissues. We hope to build in flexibility with this method so people can use different columns and dilution schemes. Not every laboratory has the same instrumentation or uses the same specific columns. Again, this is a collaborative effort among the Department of Defense, EPA Office of Water, EPA Office of Land and Emergency Management, and ORD. We're currently running a single lab validation. Assuming that's successful, we hope to follow with a multi-lab validation. This slide says summer 2020. Obviously, the pandemic is going to push that back. We hope to get the single lab validation done by the end of this calendar year and move on with a multi-lab validation in early 2021. So moving on into marine waters, we have no EPA-approved method for PFAS in marine waters. However, we do have what we call a research-grade method. It's based on isotope dilution. It covers 24 PFAS, and it is very similar to 537, 533, and 8327. It uses solid phase extraction for sample concentration, and there's another step to get rid of some of the matrix that you encounter with marine waters. It uses weak ion exchange, and we've adapted it for measuring estuarine sediments as well as for the total organic precursor assay. In very small letters at the bottom of the slide are some contact information if you have specific questions about this particular method. For fish tissues, again, we have no approved method yet. That is, as I mentioned, part of the isotope dilution method that we're single lab validating, but we don't have one yet. But a research method using LC-MS solid phase extraction and isotope dilution is being used for fish tissue right now. It covers 13 carboxylic and sulfonic acids ranging from C4 to C12, as well as PFOSA, for what totals now 33 analytes. Again, it's an isotope dilution method where you spike in the stable isotopically labeled PFAS analogs into 1 to 2 grams of fillet tissue. The sample is digested with caustic, either potassium hydroxide or sodium hydroxide methanol solution to release the PFAS. Then you separate the solids and process the aqueous solution that's left over by solid phase extraction, and then use LC-MS for analysis. Typically, for the fish tissues that we've looked at, the longer chains are usually the most persistent, C8 and above. And again, contact information at the bottom of the slide if you have specific questions about this method. Now, backing up from specific methods, I'm going to talk a little bit about total organic fluorine using combustion ion chromatography. This is an extremely high priority for the Office of Water and EPA right now, more of a priority than the total organic precursor assay. So we're in the process of developing a formalized method for what we hope will be a single and then multi-lab validation process for a TOF method. But where we are right now is that the development is just underway. Essentially, this approach really requires no specialized or costly instrumentation. You can apply it to aqueous matrices as well as blood serum. And there's also a way that you can go about analyzing solid samples directly using total ion or combustion ion chromatography with ion chromatography. The absolute key to this method is removing inorganic fluorine from the sample, and that is a lot more difficult than it may sound. And what that essentially will mean is that, did somebody have a question? Okay.
No, it was just a new person joining, sorry.
Okay, so essentially what that means is that sensitivity just may not be that low, and that is a real challenge for total organic fluorine. So, when we're talking about in aqueous samples using specific LC-MS methods, we're talking about, 1, maybe even less than 1 nanogram per liter for a lot of specific compounds. This may just not be able to get us down to those kinds of levels for a total organic fluorine assessment. The best we may be able to do is in the 100 to 10 tens of nanograms per liter, and even then, I think that's a tall order. And again, a big part of that challenge is dealing with existing inorganic fluorine in the sample. However, it could be developed on a wide commercial scale, and as I mentioned earlier, this is a very high priority for EPA. Another quote-unquote total approach, if you will, is total organic precursors. This is an approach that was developed by Houts. There are no multi-laboratory validated standard methods that we know of. However, this method is available from some contract laboratories. It does not identify individual precursor compounds. It just gives you essentially a snapshot of what may be there in your sample that is considered a precursor. And essentially what you're doing is you're analyzing the sample twice. So whatever method you're using, you analyze the sample, then you oxidize the sample, and you reanalyze that sample to see the shift in your PFAS chemical species. You can use it for aqueous and solid matrices, and it gives a very conservative estimate of the total concentration of the precursors. But one of the drawbacks, it is more expensive because you need to analyze that sample twice. So in summary, we do have validated methods now in drinking water, 533 and 537.1. We've got a validated method that's undergoing final review for non-drinking water or non-potable water for 24 PFAS. And then we are currently embarking or working on a single and multi-lab validation for non-potable water samples as well as solids that include 40 PFAS. That's going to take some time because there's so many matrices involved and so many labs that we have to line up eventually for the multi-lab validation. However, there is a chance that if the single lab validation is successful, we could post that as a draft method by the end of the calendar year. We're also continuing to develop methods in the research laboratories for fish tissue serum, ambient air, which I did not talk about, but if you have any questions about air sampling and air analyses, you can get a hold of me and I'll put you in touch with the right people. Total organic precursors, our big emphasis though on the next method is total organic fluorine and standardizing a method there. And then the high resolution mass spectroscopy that we continue to apply non-target analysis to environmental samples throughout the US. There's my contact information. Please don't hesitate to email or give me a call. And I appreciate the time, and I'll turn it over to Stefan.
Yes, thanks to OECD to make possible for me to present our work within CEN, European Standardization, and ISO. First, I mean, this is a joint work between a number of working groups, and shortly it says independent. That I retired 1 year ago, and before then I worked 35 years in textile research for Governmental Institute in Sweden, and also my background on fluorochemistry. I used to work as a drafter for the Secretariat in the Stockholm Convention on PFAS and alternatives for more than 5 years until 2016, and doing research up and down on fluorochemistry for the past around 20 years. So I'm very happy to present a bit about our work on to produce analytical methods on PFAS in textiles and leather as a joint activity in the European Standardization and ISO. Next slide, please. Well, that was just a nice illustration of different materials we look at, and this is a bit different, looking at leather and textiles than environmental samples, which is also a very big challenge, as you heard from our colleagues in the previous presentations. But it could be a bit different with these matrices. So the next slide, please.
Sorry, I'm just going to interrupt for 2 seconds. If Laura can make it at full screen, I think it'll be easier for people with small screens to see.
Yes.
Sorry about that. Thanks.
No problem.
Thank you both.
Thank you. The standardization work on PFAS that started this roughly 15 years ago, the European REACH legislation. We had this directive, the restriction directive for PFAS, and I think were 8 PFAS substances. And that was a work started in roughly 2005, 2006. And came up with a technical specification for 8 PFAS substances 2010. And the technical specification should have a lifespan of 3 years plus 3 years. And this method that came up then, the CEN TS 15968, is still used around the globe for the analysis of PFAS, but it was originally used to analyze PFAS and some other PFAS-related substances. Next slide, please. Yeah, thank you. And these are the substances that were, until from then, there have been some activities in different working groups in the European standardization. And here is an example for the leather working group, the TC289. They started and actually presented a test method for a number of PFAS in 2016. I think it's a published standard today. Roughly 8 substances, all acids, perfluorinated acids. We saw after some activities, we have these joint meetings between the working groups on leather and textile, that we need to do some common work on the development of PFAS, because there were separate activities going on in different areas within the European standardization organization. Next slide, please. So in 2018, we did a number of groups, as you can see here, from textile and leather and footwear, decided to have a joint activity to share experience, to have conformity on PFAS substances that are relevant for those materials and those products. And also, of course, to have a common ground on methodology and the testing procedure, preparation of samples, etc. So we decided then to have a joint work to find common ground to develop standardized test methods for PFAS for these materials. Next slide, please. So we first of all, I mean, the procedure for developing a standard or a test method, whatever, are, as you can see on the screen. So our focus in this work was to fix a list of relevant for textile and for leather relevant key PFAS substances. You saw in the earlier presentations, there are more than 4,700 CAS numbered PFAS substances, and to find the absolute high relevant PFAS in this vast number of PFAS substances is really a challenge. So, and also, besides to find the key relevant PFAS substances, also to define consistent analytical conditions for more reliable results and more reliable test methods. Next slide, please. So we started, the first step was to categorize groups of PFAS. This table is a result of work with in the Stockholm Convention on PFOS and the PFAS, non-PFOS also non-PFAS, but PFAS alternatives, where different categories and subcategories of PFAS were related to the uses of PFAS depending on which subcategory, meaning which the hydrophilic end group was presented on this PFAS molecule. And so we decided to put them in letter categories, as you can see in the right side of this table, and address these to the relevance for leather and textile, which are presented in the next slide. Where in this slide you can see that PFAS polymers that represent the category H are likely relevant, and also I for textiles and leather products. So we on those these PFAS that have reference substances for quantitative analysis and relate to these categories could be addressed in the coming standards. Next slide, please. So we have a long list of key PFAS substances that have some radioisotope reference substance, and also they are represented in the European legislation and also global in the Global Stockholm Convention. And we tried with this table to address them to the relevance for footwear, leather, and textiles. Both to have an intention to cover regulatory requirements, but also relevant requirements for those materials we were looking at for the coming standard. And we also categorized these substances, as we had earlier. There's the volatile and non-volatile PFASs, whether they were applicable for liquid chromatography or gas chromatography, or sometimes both. So we have this table, which continues in 2 more slides. It's a starting point for relevance and applicable test methods for these standards. Next slide, please. We can see here further on that we separated them in the perfluorinated sulfonic acids, sulfonamides, sulfonamide ethanols, and we categorized them depending on and we also addressed how they appear, contaminant or intended use, what functionality they possibly have, and categorize them depending on their functionality. Next slide, please. So we identified that these H labeled chemicals had high relevance to textiles and leather, but also we have to include PFAS, especially perfluorinated acids, that could appear as contaminants in these materials as well, and of course in context to regulatory requirements for a number of these substances. Next slide, please. So what we are going to do, and we are doing at the moment, is to put these 36 identified key PFAS substances for this method to look at their details on analytical techniques, characteristic IMMs, characteristic ions, internal standard, and to look at detection limits for each and every relevant PFAS standard. So this is ongoing work to set up the testing method on these identified relevant PFAS for leather and textiles. And this will be done in these 3 separate groups, with the leather, footwear, and textiles, but with this common experience and to work further with to develop conform standards on these materials. Next slide, please. So we are separately now working on these procedures to develop the actual standards, and in the textile working group, which I'm a part since many years, we have a preliminary European standards on the table for inquiry, and since they are labeled 2020, we hope that they will be out published in this year. And there are two parts in this standard. There is one part for the non-volatile and one part for the volatile PFAS or neutral. And we hope that it will be possible to publish these standards also, but that's maybe my wish also, as a European standard, an CEN and ISO standard is, when it's out there. Next slide, please. So conclusively, we are in different phases in the formal process to present these standards. As I mentioned, for the working group 26, which is the textiles, we are close to inquiry for this standard I mentioned, and the other working groups, they're also on the way, but I since I'm not directly involved in this work, I will not exactly detail where they are in this formal process, but we have a common ground on the relevant PFASs and procedures. So I think we are quite even here on the standardization process. So conclusively, we have at the moment we have a common list of PFASs that are compatible with the European legislation, and also what's going on a wider scope of regulation in Europe, in the European Union on PFASs. But of course, with the test method the test methods that are of this kind, we cannot and will probably not ever cover all kind of PFASs, but we at least have, as we're quite sure about that, 36 key substances, so we can say something about these coming test samples. The intention with these standards when they are published, and of course until they are published, is to have this transparency and publicly available test methods, because we have seen that there are quite a number of activities around the globe where you have their in-house standards, you have different test methods with good knowledge after at least 15 years having worked with this in a number of different areas, but to make consistent test methods, so we have reliable test results you can compare from different laboratories. And of course, when the market has more and more requirements in textile and the leather market, in this case on PFASs, they need good tools to compare and to eventually do substitution to understand their value chains and the possible use of PFASs in their value chains, and see whether they can be substituted or not. So by the end of this slide, you can see the conveners of these working groups, and the last row is me. So if you want additional information of our work, you're absolutely welcome to contact either of us or all of us for further detailed information. Thank you very much.
Thank you, Stefan, for the presentation, and indeed, a big thank you to all three speakers for this comprehensive overview. The first two focusing more on different environmental media and the methods being used there, and the types of PFAS being targeted, as well as some of the challenges and developments. And then the final presentation taking a look at specific product types, textile and leather. I think we'll end today's webinar now. I'd like to again take this opportunity to thank all of the three speakers for sharing their expertise and experience with us, and invite anybody who has suggestions for specific topical webinars that the Global PFC group could host to get in touch with the OECD Secretary.